My name is Kruzer, and I worked in the medical laboratory internship at Maine campus this summer. I'm a rising sophomore at Case Western Reserve University. My research study focused on a comparison of an immunochromatographic rapid test, a microplate enzyme immunoassay, and traditional culture methods for detection of Campylobacter species in outpatient stool samples. Campylobacter is a leading cause of bacterial gastroenteritis in humans, affecting over 2.4 million persons each year in developing and developed countries globally. Campylobacteriosis is a foodborne illness. It's an infection that is usually caused by consuming unpasteurized milk, contaminated food or water, or undercooked meat and poultry that results in diarrhea with varying severity from loose to bloody stools. There are multiple current detection methods, which include recovery by traditional culture techniques, immunochromatographic rapid tests, and enzyme immunoassays for the qualitative detection of Campylobacter-specific antigen in human stool. Campylobacter is a microaerophilic, gram-negative bacillus that grows as a flat, gray, watery colony on solid agar. An analytical review of recent publications suggests a problem with consistent detection of Campylobacter. Therefore, a comparison of antigen detection methods versus culture techniques needs to be conducted. There were three main objectives for this study. The first objective was to compare two antigen detection methods to the recovery of Campylobacter species in stool cultures. The second objective was to tabulate the incidence of bacterial, parasitic, and viral stool pathogens in outpatients who present with diarrhea. The third objective was to compare the sensitivity for recovery of Campylobacter species from stool specimens using culture and two antigen methods. For this study, 100 stool samples were collected from outpatients and transported to the lab in Carrie Blair preservative. 3 diagnostic methods were performed on each sample for the detection of Campylobacter species. All samples were stored at 2 to 8 degrees Celsius and tested within 3 days of collection. I'm going to start by discussing the first assay I used, which was ImmunoCard Stat-Campy. This is an immunochromatographic rapid test that detects Campylobacter antigens in human stools. 350 microliters of the sample diluent was vortexed with 50 microliters of the stool for 15 seconds. Then, 175 microliters of the diluted specimen was added to the device sample port. A positive test was indicated when a red band appeared at the control and test line after a 20-minute incubation period, which indicated the presence of the antigen. Figure 1 depicts the immunochromatographic test. The left image shows a positive test due to the red band at the control and test lines. The right image depicts a negative test since the red band is not present at the test line. Next, I'm going to discuss the second testing method, which was the traditional culture method. 2 variations of culture method were performed. For the selective culture, 1 drop of fecal solution was inoculated directly onto Campylobacter selective medium and streaked for isolation. For the filtration culture, 10 drops of fecal suspension were dispensed onto a 0.45 micron pore size filter on 5% sheep blood agar plates, the BAP. BAPs were incubated for 1 hour, enabling the Campylobacter bacteria to wiggle through the filter. Then the filters were aseptically discarded. Both the Campylobacter selective medium and the BAPs were incubated in a microaerophilic atmosphere at 42 degrees Celsius. They were incubated with a Campylobacter gas generating system for 72 hours in an enclosed jar. The plates were then examined for the presence of small, gray, watery, non-hemolytic colonies. Campylobacter species were identified through multiple tests, including a gram stain, catalase and oxidase, hippurate hydrolysis, motility test, and a disc diffusion antimicrobial susceptibility tests. Campylobacter was observed to be a seagull-shaped gram-negative bacilli, catalase and oxidase positive, hippurate positive, and it had darting motility. Susceptibility to nalidixic acid, cephalothin, and erythromycin identified the species of Campylobacter. For this, the bacteria was resistant if there was growth, and the bacteria was susceptible if there was a zone of inhibition. Any positive result by the EIA or culture was considered a true positive and directly compared with the immunochromatographic test. Therefore, the gold standard was twofold. This slide shows images from multiple testing procedures. The left half of Figure 2 depicts a specimen dispensed onto the filter on the blood agar plate. As you can see, 10 to 15 drops of the specimen was dispensed onto the filter, which was aseptically removed 1 hour later. The right half of the image illustrates the growth of Campylobacter jejuni on the blood agar plate. Figure 3 shows a hip rate hydrolysis test, with the first 2 test tubes being the positive and negative control. A positive reaction is evidenced by observation of a dark purple coloration, as seen in the first, third, and fourth test tubes. Now I will discuss the final testing method, which was the Prospect-T EIA test. This is a solid-phase immunoassay for the detection of Campylobacter-specific antigen. It is a testing procedure that the Cleveland Clinic Laboratory uses as its routine method. For this test, the diluted stool samples were added to breakaway microplate wells on which the antibody was bound. The wells were incubated and washed to remove the unbound specimen. In a positive result, the enzyme conjugate was bound to the well and resulted in a yellow coloration, whereas a negative result was colorless. This testing method takes approximately 2 hours, and there is risk for crossover between the wells. Table 4 shows a comparison of Campylobacter species detected by each testing method and the length of time for detection. The immunostat, EIA, and selective culture all grow Campylobacter jejuni and Campylobacter coli, whereas the filtration culture grows each type of Campylobacter species. The immunostat takes approximately 1 hour for completion, the EIA takes 2 hours, and the culture methods take 72 hours. Figure 5 shows a table of discordance with 100 samples. The majority of tests yielded a negative reaction, thus indicating a negative detection of Campylobacter. Figure 6 depicts the total number of positives detected by EIA, the immunostat, and the culture methods. The EIA and immunostat detected 7 positives, whereas culture and filtration detected only 3 positives. Figure 7 shows the incidence of bacterial, parasitic, and viral stool pathogens in outpatients who present to their clinicians with diarrhea. Campylobacter was the most prevalent enteric pathogen identified with 10 positives, followed by C. diff with 9 positives. 2 cases each of salmonella and parasites were identified, and there were 0 cases of E. coli 0157, Shigella, Shiga toxin, and rotavirus. Figure 8 depicts the sensitivity, specificity, and predictive values when culture served as the gold standard. The EIA had a higher sensitivity, specificity, and positive predictive value, along with negative predictive value, than the immunocardstat CAMPI, which detected only 2 of the 3 positives. Figure 9 shows the sensitivity, specificity, and predictive values when the EIA and culture served as the reference method. In comparison to when culture only was the gold standard, the sensitivity decreased, but the specificity and positive predictive values increased. Therefore, the immunostat test is a very good test for correctly identifying negative results. In conclusion, when comparing the 2 antigen detection methods to the traditional culture methods, the antigen detection method recovered 7 positives, whereas the culture recovered only 3 positives. Perhaps some of the discordance can be attributed to false positive and false negative results. The sensitivity, specificity, and predictive values were also calculated with a 2-fold gold standard. When the culture served as the reference test, the sensitivity and specificity were relatively high, both being greater than 66%, whereas the positive predictive value for the immunostat dropped to 28%. When the positive EIA or culture served as the reference test, the immunostat sensitivity decreased, but the positive predictive value and specificity increased. The highest incidence of bacterial pathogens was Campylobacter species and C. diff, followed by 2 cases each presenting with salmonella and parasites. Consistently reliable identification of Campylobacter species is crucial for the diagnosis of the leading cause of human enteritis globally. In the future, it's highly recommended to continue this study with a larger sample size, which can validate the findings on a broader scale. For a larger volume of positive Campylobacter, It may be beneficial for community sites to share their specimens with the main campus laboratory. It's recommended to follow any positive EIA or immunostat with a traditional culture. A PCR may enable highly sensitive and specific detection of all Campylobacter species and can perhaps be considered the new gold standard. Periodic assessment of the reliability of laboratory procedures is highly recommended. And I'd like to give my special gratitude to the Cleveland Clinic Microbiology Laboratory, along with my mentor, Dr. Hall, and Suzanne Schroeder, Deborah Warner, and the Office of Civic Education Initiatives.